This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Hi everyone, this is Audiommunity, recorded on April 5th, 2016. I'm Kevin Bonham, and I'm joined by Kate France.
Hello.
Who apparently was recently in the Boston Globe because she's trying to unionize, and the man wants to keep her down.
Power to the people!
Right. Grad employees, unite! The only thing you have to lose are your chains! Okay, it's not that political, but...
And the other voice you hear is Matt Woodruff.
Hello, everyone.
And it's been a while since we recorded, and I've been delinquent in posting stuff, so I still haven't posted. We spent some time with Jesse Knorr of Bacteria Files, and I'm going to edit that and post it before this episode posts. But, so that's going to go up, and then we recorded something several weeks ago that I also haven't edited, and I don't know if I'm going to edit. I might just put this one out first.
I was going to say, by the time you hear this, it already was done.
Yeah, right. Yeah. No, I think I might actually post this so we can, get caught up in time so that we're actually, recording close to the time we post things.
Agreed.
But we have another episode in the doc, in principle, if I can just get my shit together and put it up. Today, we're going to be discussing a paper that's a little bit different than our usual fare. It's still immunology, but it's this really weird paper. It's a case report of a guy who got a bone marrow transplant and got an allergy from the bone marrow transplant, which is some really cool immunology. Sucks for this dude, although I suppose it's nice that he was able to get a bone marrow transplant. Right.
Yeah, so it might be worth talking a little bit about how we select these papers sometimes, so people might get the false impression that we're doing this intelligently and that we're somehow ranking these papers as we think that they are important to the field, and that's really not the case at all. Usually the way this works is someone will be trolling around on the internet and someone will say, kiwi allergy, that's cool, and that's pretty much what happened here. So I don't even know that this is a spectacularly relevant paper for the broader view of immunology, but I do think it involves kiwis, and that's fun.
Yeah, kiwis are delicious.
I think that this is a good paper to talk about some basic allergy questions. I have a lot of questions that aren't maybe related to this paper directly, but have to do with the establishment of allergy. So right. I think this will be good. So
yeah, that's fair. And,
don't hit
and
skip.
No, I think it'll be a fun discussion, if nothing else, because it's about kiwis.
Right. Okay, so let's back up for a second and say, what are we drinking? Because it looks like Kate is at work. And
I'm at work.
Damn you, Kate.
I know.
Working too hard. You really need to get this union going so that you can leave at 4 p.m. every day.
I know.
And not have to do any work.
Get those rights protected.
How does John feel about the union push?
I haven't asked him. The faculty support has been interesting because Harvard's been meeting with them and asking them to not be, the administration has been meeting with faculty and asking them to not be outwardly supportive and question the motives of the union to students and highlight the this all came out on the memo got leaked, but highlight the disadvantages of forming a union.
Yeah.
So I think right now the idea is that students who don't directly work with the faculty will approach the faculty and talk to them just to keep it from being really awkward.
Okay.
Right.
I see.
So I don't know. John at one point said, I don't think I can talk to you about this, and walked away. And I didn't really follow that up because I was too busy doing actual work. So.
Right.
He's like, I want to get tenure someday, and so I'm not talking to you about this.
That's the other thing. We're only asking support from tenured faculty members.
Oh, right. That's probably a good plan.
Yeah.
Yeah.
Okay, so leftist politics aside, so what are we drinking? Matt, maybe?
I actually am not drinking anything yet, but I am planning on drinking kiwi juice later because I've got 6 kiwis in the fridge, and that'll be my celebration.
That's really mean.
Yep.
Well, I guess, again, I am left holding the ball. I actually decided to go to the liquor store today to get my own beer that I could keep in my office just for this purpose. And so I got some Cisco Brewery Grey Lady, which is sort of like Earl Grey, because it's got bergamot in it, and it's really delicious. It's like, if you like Earl Grey tea and you like beer, you will like Grey Lady from Cisco Brewery.
Grey Lady is pretty good. I don't like the idea of tea being in my beer, though, so I sort of object to the idea of that. Although I have had the beer and it's delicious, so I'm finding it difficult to object to your beer.
Yeah. There's just nothing wrong with this.
Okay.
I mean, I can see how, like, from a sort of like a priori, you would think like tea and beer, that's gross, but it just happens to be delicious. Yeah. And I'm probably going to be drinking this for the next couple of weeks because I don't intend to drink like 6 of them tonight. So
it's going to be a really spirited discussion.
Right. So the paper that we're discussing today is called Newly Acquired Kiwi Fruit Allergy After Bone Marrow Transplantation from a Kiwi Allergic Donor. And like, really, that's the whole paper. It's just right there in the title, which is nice. The first author is Gerzors. And the last author is Irish. And it's in the Journal of European Academy of Dermatology and Venereology?
Yeah, that means
venereology.
Venereal disease?
That would be the study of sexually transmitted diseases.
Right. This is not a journal that is on my like weekly reading list.
Oh, but it's a fun time.
But this is a pretty cool paper. I think I might have found it on Reddit, if I'm being perfectly honest. But basically the idea is that you have a guy who's got a myeloma, and he needs to get what's called a myeloablative hematopoietic stem cell transplant. So they're going to wipe out his bone marrow. This is a cancer of some bone marrow derived cell. So we're going to wipe out his bone marrow.
Yeah, I think it's a lymphocytic leukemia too. So it's either B cells or T cells.
Okay, my bad.
Yeah.
It's called myeloablative hematopoietic stem cell transplant, but it's for all kinds of immune cells. So maybe we haven't talked about this, and it's sort of assumed, but in immunology, we like to sort of divide the body up into 2 things. There's the hematopoietic stem cell niche, which is anything that derives from bone marrow stem cells. So that's red blood cells, all of your lymphocytes, all of your innate immune cells, basically everything that's associated with the immune system, red and white blood cells. And then there's everything else.
Right, and we call that stroma.
Right. So
it's
the reason
maybe immunocentric. I mean, the argument could be made that, we're not seeing the whole picture, but I like stroma.
Sure, but I think the reason for this separation is purely historical and based on the methods, because if you irradiate an organism, the thing that dies the most is the hematopoietic stem cell niche. So we can irradiate an animal, can basically wipe out its bone marrow derived stem cells because they're constantly regenerating. And then the other thing, part of that though, is that we can completely replace the hematopoietic stem cell system with a stem cell transplant, with a bone marrow transplant. So this has been really useful in immunology. You can sort of like take a mouse, you can give it the immune system of a different mouse with different genetic backgrounds, and you can sort of test a bunch of things. And so historically, this technique has been used a lot to really great advantage. It also happens to be the case that we can do bone marrow transplants in people, and I think we've talked about that in the past. But it turns out that if you have a bone marrow tumor, in other words, a cancer that is from a cell that is of a hematopoietic lineage, then you can sort of wipe out the bone marrow and put in new bone marrow, and that will often treat the disease with some complications, of course.
Yeah, some major ones.
Yeah, right.
Which we've talked about.
Right,
which we have talked about in the past. So this guy has a cancer. They do this radiation treatment, wipe out his bone marrow, and give him a bone marrow transplant from his sister, who is an HLA match. And so he gets bone marrow from his sister and now has a completely new immune system. So all of the T cells are new, all of the B cells are new. There's probably some like macrophages and dendritic cells in the tissue that are from his germline, but most of the cells of his immune system are newly derived from this transplant. And he has a kiwi allergy that he never noticed before. And that's sort of where this paper starts. So, go ahead.
Oh, I was going to say, what's important to know is that his donor, his sister, had the kiwi allergy since early childhood.
Oh, right. Yes.
So, since he had this like late onset kiwi allergy that came when he received the bone marrow from his sister, the hypothesis was perhaps this kiwi allergy has to do with the cells that were transmitted. So maybe the bone marrow maybe the bone marrow transplant also transplanted this kiwi allergy.
Right. And I think it's worth saying maybe that allergies in general they're not genetic in the way that you might think of other genetic diseases. So you might have a predisposition to allergy, but the specific allergy that you get is because of a particular VDJ recombination in your B cells. Maybe we should get into the biology of allergy a little bit.
Sure.
So if you recall, I think we talked about this in one of our very early episodes, but one of the hallmarks of the mammalian immune system is our T and B cells have this really cool mechanism to generate a diverse receptor repertoire, where they actually rearrange their they rearrange particular genes at the DNA level, actually cut up and rearrange the B cell receptor, the T cell receptor locus, and that happens in a sort of random fashion. So everybody has a different set of B cells, everybody has a different set of T cells. And so if you have a particular allergy, I'm not describing this well.
No. Okay, so you have these B cells and T cells, and they're specific for different antigens. So the T cell receptor is going to bind particular peptides. You're hoping that you're going to get a bunch of T cells that are going to recognize virus peptides and bacterial peptides so you can clear infections, but sometimes they recognize harmless peptides. And usually these sorts of recognitions would be deleted because there's a selection process for the T cells. But it's not always the case. So
So I think that it's a little bit different in the case of allergy. So I think what you're getting at is
antibodies.
Yeah.
Well, I got hopped on T cells, and I'm like, I need to bring this back to antibodies.
Yeah. So take 3, maybe, I suppose. So definitely diverse receptors on both sides, right? You've got a T cell and a B cell receptor, and so we've talked about.
I guess the point that I wanted to make though is that just because this person has a particular antibody against a kiwi, that doesn't mean that someone that is like, there's no reason to assume that the brother, sort of from a genetic background or from a genetic standpoint, would have the same antibody, because antibodies are randomly generated and T cell receptors are randomly generated.
Right.
So there's no reason to think that this particular B cell or T cell response would be the same, even if you had identical twins.
No.
They'd have a completely different B cell and T cell repertoire.
Right. Now, the thing that we want to, I think, stress, though, in allergy is that something has gone wrong, right? So allergy is not a normal occurrence in the immune system. And so we've talked about autoimmunity, and I think that this is sort of what Kate was getting at before, where you have sort of the aberrant activation of T cells and B cells that react against self, right? And so when you have T cells and B cells that react against self, when they first develop, they test themselves, they test their specificity against lots of different proteins that are in your body. And so anybody that is specific for yourself is deleted, centrally or peripherally, and we've talked about that in the past. In the case of allergy, these B cells and T cells might not necessarily have ever been deleted, right? So in kiwi, for example, there's presumably some protein in kiwi that you don't have as a human. That's normal. And so you might expect that you'd have B cell precursors that would normally respond to kiwi. Just random reassortment, you're going to have some B cells that respond to kiwi. So the real question in allergy is not so much why the B cells and the T cells that respond to kiwi are there to begin with. The question is, what about that kiwi protein in this one particular person made your immune system think that kiwi protein was dangerous? And so when we're talking about allergy, what we're really talking about is some aberrant danger signal that caused you as an immune system to decide that a foreign protein that you may or may not make contact with every day, in the case of like a pollen allergy, for example, why is it that you decided this pollen, rather than being just an inert part of my daily surroundings, why is that dangerous, and why do I feel the need to respond to it every time I see it now?
Yes.
Is that fair?
I agree.
Yes.
All right. And so allergy in particular, when you get it, there is this specific type of response. And it's, to be honest with you, it's a little unclear to me why this would be the case. But for some reason, when you respond against something you're not supposed to, in the case of an allergy, you tend to respond as if that thing was a parasite.
Yeah.
And so we've talked before about how the immune system responds different ways depending on the pathogen it's trying to fight. And the way that you fight parasites, large organisms that are infecting you, is creating this really mess of a toxic environment around the parasite, with things like the release of histamine and proteases and all of these things that just are not good for parasitic life to persist. And so when we're talking about like antihistamines that we use to treat allergy, what we're really doing is we are trying to dampen down this anti-parasite-like response to a protein that otherwise is totally innocuous and is not a threat to you in any way.
Yeah, so I think there's an interesting piece of what you just said. So why the fact that allergies tend to have a particular phenotype, in other words, they have this anti-parasite immune phenotype, and the fact that we don't have allergies to anything. Like, I've never heard of a steak protein allergy, for example.
Yeah. We have allergies.
We have allergies to very, very specific things. So
things
pollen is a very common one. Right. Dust mites is a common one. And as Kate just said, they tend to be things that have proteases. Right. And proteases are a type of enzyme that cuts proteins, and a particular type of protease, specifically cysteine proteases, right?
Yeah. Pollen, kiwi, papaya, pineapple, those are all like really common allergies. They all have these proteases. I always wonder, has anyone ever found like the establishment, can you make a mouse allergic to something?
Oh yeah. Sure you can.
Well, you can
Sure you can. You can take house dust mites and you can mash them into bits and you can make the mouse inhale them over and over again. That totally works.
You inject them with you don't inject them with CFA. You inject them with.
No, they just inhale them.
I remember we had this as a paper.
Yeah.
But could you take like antigens and just like make them protease dead and see if you would still get allergy? Obviously, you can't activate it with anything. If the allergy is dependent on like protease damage, the food protein being there, and then like something else, like a particular bacteria or something, you would never be able to recapitulate it. But could you just so you can't use like CFA, but could you just make them allergic? Or try to make them allergic with like protease dead or protease active?
I'm nearly positive that our colleague in the immunology program, Dan Dwyer, did that experiment with papain, which is a protease from papaya. It's often used experimentally. And I may be getting this wrong, and so if Dan listens to this, he's going to give me shit for it. But I'm pretty sure that very early on in his PhD work, he was using papain and the papain dead, or the, like protease dead papain, and looking at differential responses. And unfortunately, I don't remember.
Yeah, you know what's sad is
what's induced allergies or not. What's
sad is I'm the second author on that paper, and I have no idea.
Good job. Okay, good job.
Yeah. Well done, Matt. Yep. All right, we are good scientists. Yep. The point is that people have thought of that. I suspect that somebody knows the answer. We should really go look that up.
Yeah.
Jesus.
Yeah. Also,
we should look that
crabs.
up.
So I was looking at the list of allergies that the donor has. So it looks like they also have crab and carrot allergies. So I assume that there are proteases in those as well. But what's super interesting to me is I imagine they must have tested the recipient. You know, if you're going to do this case study, you would have them eat kiwi and crab and like all the things. It looks like he only got the kiwi allergy.
Mm-hmm. That doesn't surprise me, actually. So we should take another step back and talk about what it is that is actually causing allergy. So this worm-type immunity that Matt was alluding to, this Th2-driven immunity, one of the hallmarks of it is that the B cells that are generated during a Th2 response are B cells that have undergone isotype switching to IgE. And have we talked about isotype switching before?
Yeah.
On this podcast? Okay. So just for a brief recap, basically antibodies come in a bunch of different flavors. And basically they have the antigen binding region, and they also have what's called the constant region. And the the antigen binding domain is what determines whether the B cell gets activated. But then the B cell is able to swap out the constant domains, which may seem like sort of a poor name. If it's constant, it shouldn't be switching. But in any case, the constant region shifts out and can have different effector functions. So you can have the same antibody binding, the same antigen binding domain and a different constant region, and the antibody will have different functions. And so the function of IgE is to be bound on the surface of a bunch of different cell types like basophils and eosinophils. And so these cells are decorated with lots of different IgE molecules. And so if they bind to their antigen, that causes degranulation of these cells and the release of all these chemicals that Matt mentioned, like histamines and a bunch of other crap.
Right. So you can, and I think we've used this analogy before, but you can think of them as like little cytokine bombs. So the effector cells that we're talking about, basophils, mast cells, eosinophils, all these guys, they have these physical granules. You can see them under the microscope, these little tiny granules in their cytoplasm. And those granules are filled with a whole bunch of stuff that is very, very inflammatory. So mast cells, for example, will release a ton of TNF alpha almost immediately, within minutes of stimulation. And they do that by just sort of opening these granules out into the world. So you can imagine them as these super sensitive cytokine bombs. The thing is that they don't have a trigger in and of themselves. So, there's some thoughts that they could sense some of the damps that we've talked about, some of the different TLRs, but the most studied way, at least, that they sort of identify when to blow up is they rely on these antibodies that are just sort of floating around in the organism's serum. And so if your B cells have responded against pollen, and you've produced a ton of IgE, those IgE-specific antibodies will just sort of sit on these cytokine bombs, and they can detect whether those antibodies see pollen or not. So it's almost like a surrogate B-cell receptor, right? And so when pollen comes along, mast cells decorated with anti-pollen IgE get tickled, and all of a sudden you have TNF and histamine and IL-1 beta and like all of this stuff that just sort of explodes out into the environment.
Right, and
I have to head out, you guys. Sorry.
Okay, Kate has to leave us. That's fine. I was really looking forward to your... Do you have any questions about this paper, even if we haven't talked about...
Yes.
Because you had mentioned you had some questions. I do have questions about allergies.
I have a question on how... Well, I have lots of questions. But a couple questions I have are, do people think allergies are like just that you make the response against the wrong protein, but that there actually was some sort of damage and like immune function, like immune action, and like your allergy protein just like got caught in the middle? So it was that you ate kiwi and then you like got infected and you made a response against kiwi? Or is it that like you got infected with a weird virus and then you created a response against something that looks just like kiwi? And then my other question is, if immune responses take like weeks to develop, why are allergy responses immediate? Some of these I know the answer to.
Okay.
All right. So I wrote it down, so I will bring them up when it becomes appropriate.
Okay. Those are some of my questions. You're welcome.
All right. Cool.
I'll see you guys later.
See ya.
Bye. Okay, so let me go back. What was I saying? No, I lost it.
Okay, well, so we were just talking about cytokine bombs and...
Oh, right. So, okay, yes, I remember now. So the idea of these cytokine bombs not having any receptor of their own, like a TLR or something else. What they do have is they have a receptor called Ig epsilon. I think it's receptor 3, maybe?
Yeah.
So it's a high affinity receptor for the constant region of IgE.
Right.
So this is kind of a brilliant idea. You can have one cell that expresses one type of receptor, but it can then gain the ability to bind to a bunch of different antigens depending on what the organism has seen in the past. So you have B cells that make these IgE antibodies, and then they get sucked up by all of these cells expressing the IgE receptor, and then they just hang out on the surface of these cells. And actually, there is very, very little IgE present in serum because it just gets soaked up by all of these effector cells, these like basophils, eosinophils, mast cells that express the high affinity IgE receptor. So they basically use the IgE receptor as their pattern recognition receptor, as their antigen receptor, except the receptor itself sees what the antibodies that are in that particular organism see.
Right.
I don't know if I made that make sense, but I think it's this really cool evolutionary strategy where you have one cell that basically produces the receptors that another cell soaks up and then is able to use.
Right. Yeah, no, I think it's a good explanation. I have a theoretical question for you.
Sure.
So let's assume for a second a magical animal that I give a pretty bad allergy to. So we get a spike in IgE response.
Yep.
And so now let's imagine that animal only has one allergy. And let's assume that animal does not produce any IgE at baseline. So the only IgE in this animal is the anti-allergen IgE. Okay?
Okay, so what you're saying is you have one or more B cells.
Yep. That are produces
an IgE isotype antibody that is specific for your allergen X.
Right.
Yes.
But that is the only IgE in the serum. Okay.
Mm-hmm.
Now let's imagine that I went through and found a way to eliminate every B cell and IgE molecule in that mouse. So I wipe out all the memory B cells, and I wipe out all of the serum antibody, and I wipe out all of the antibody off of the surface of mast cells and basophils and eosinophils. Okay?
Okay.
And then I give that animal that allergen. What happens?
You said you wiped out the memory B cells as well?
Memory B cells as well. Memory B cells are gone. So any humoral responsiveness that remembers this thing is now gone.
So I think that you basically get a naive response. So
here's,
as if that organism never had a B cell or never had the allergy in the past. So I think it's agreed that allergies have to be pre-sensitized. Like you can't have a bee venom allergy if you've never seen bee venom.
Yeah. Also, right? Jenny McCarthy, by the way, just started the, I think it was Jenny McCarthy. Who's the crazy one?
I think she's one of the crazy ones. She's one of the anti-vaxxers.
Yeah, I think she just started a bee venom treatment. So I guess cool. general public service announcement. If anything you do is advocated for by Jenny McCarthy, stop doing it. I think that's fair. Don't do it. Okay, so moving on to this. So here's my thoughts. I don't think that that's what I would expect. What I would expect is you would definitely get the allergy again, but you wouldn't express symptoms of the allergy the first time that you gave that mouse the allergen. Because where I think the memory of an allergy is actually going to lie, I certainly think that there's going to be some in the B cell compartment, but I almost wonder if the reservoir for allergy isn't in the T cell compartment, where you have a pre-activated T cell that at some point, for some reason or another, started producing a response against this allergen protein. And when it did that, started producing IL-4, IL-13, IL-5, like all of the cytokines required in order to get B cell differentiation towards an IgE phenotype. And so I wonder if in that mouse, what you'd actually get is a robust T cell response, the production of B cells that produce IgE, and no obvious clinical allergy response because the IgE hadn't been produced yet. But I think if you went back into that animal and you tried to sensitize them a second time, you would get all of the normal allergy response.
Okay. I think that may be true, but in order to get the allergic response, you still need to have a B cell with the correct receptor.
Sure, but you're always going to have a precursor B cell that can respond against any of these proteins, right? You and I have them floating around all the time. The question is whether they're going to get the signals in order to robustly proliferate and produce IgE.
Okay.
And so the reason that I bring it up is because when we're talking about this bone marrow transplant, so I think that this is an interesting case study, obviously, because what they were really trying to do was they were really trying to transfer hematopoietic stem cells. Right. So they really needed to reconstitute this guy's immune system. So they went to the sister and they said, your immune system's pretty close to his. There's a lower likelihood of graft versus host disease, which you've talked about before. Would you give him the transplant? So in an ideal world, they ablate this guy's cancer. It's gone. And then they go into the sister and they pull out bone marrow and they sort cells with 100% purity. And they take all of the hematopoietic stem cells and they pluck it from the sister and put it into the brother. And those cells grow out, and now you have a new immune system. Yeah?
And we should say that if that works, then you wouldn't have even had to worry about graft versus host disease.
Right.
Because all of the new T cells that would come out of that completely purified bone marrow, all of the B cells would have to first develop and go through the same processes of negative selection that all of your own immune cells go through.
Right.
From infancy till adulthood. So if you could get a completely pure hematopoietic stem cell pool, graft versus host disease wouldn't be an issue.
Right. And similarly, I think in this case, because you just mentioned allergy is not a genetic disease. You would not get an allergy to kiwi based on your germline DNA. So if we were just taking hematopoietic cells, which don't have B cell receptors, they don't have T cell receptors, they're going to turn into B cells and T cells that have receptors that could respond against kiwi, but they don't yet, which means that a pure hematopoietic stem cell population would be completely agnostic to a potential kiwi allergen, right? So really the question is, what outside of the stem cell transplant is actually causing this disease? And I think that that's where the interesting part of this really lies. So one could imagine, for example, if there was contamination of antibodies in the hematopoietic stem cell transfer. I don't know how they do this. I honestly, I don't know the procedure, and I'd be really interested. So if any listeners have the procedure where you actually pull, I'm sure we could find it. Maybe we should do that too. But
probably.
But if there is serum contamination, for example, if some of the sera from the sister went into the brother, one could imagine that those soluble IgE that have been produced by B cell responses against the kiwi could have found their way onto the brother's mast cells, basophils, all those guys, right? And so you could just have, but then you'd expect...
I suspect it would get diluted over time.
I agree with you, and also you'd expect that crab and carrot allergens would be similarly problematic. Right,
potentially, although it would be a dose thing, right? So depending on how many antibodies are present, in the serum. But the reason I say it should get diluted is because these antibodies don't live forever. There's some amount of turnover. I don't know if there's a lot of turnover on the surface of these cells themselves, but certainly there is some half-life. And once those receptors are activated and those cells degranulate, I suspect that those antibodies are spent. And so if this guy continued to consume kiwi and it was just antibody that came through, I would suspect that it would get less severe over time.
Would you expect that degranulation event, that the basophil mast cell response against kiwi that first time, even if it was just serum contamination, would you expect that could then drive an allergic response?
I don't think so. I don't think so, because that degranulation is going to be occurring not in a lymph node, not in a Peyer's patch, but it's going to be occurring somewhere that you're not going to get B cell and T cell activation, right?
What if you get a dendritic cell in the area, right? So what if you get degranulation, release of pro-Th2 cytokines, dendritic cell activation because of foreign protein plus all of these DAMPs that are getting produced as a result of the mast cell degranulation, basophil degranulation. DCs pick up the protein, which, to be fair, is foreign, and then head over to the lymph node with all the DAMPs that would be indicative of a worm infection, right? Because you're basically relying on the same sorts of danger signals that you would see in the case of an active worm infection. What if you're getting instruction of DCs out in the periphery and then presentation to T cells?
Well, you're not, though. I mean, the cytokines that are produced are not sufficient to drive further differentiation. So, well, I mean, they're in principle not supposed to be. So in other words, like, if a macrophage sees LPS, it's going to produce TNF, it's going to produce a bunch of inflammatory cytokines, and that TNF can go activate a DC, but the TNF doesn't provide the same spectrum of signals as an actual PAMP does. The cytokines themselves, if you have sterile inflammation, that's a different situation than the actual PAMP itself. That said, the idea of allergies is that for some reason that protease activity is itself a PAMP, and we don't actually know what the PAMPs are for Th2 immunity. So it's possible that in the presence of that kiwi protease, you get some inflammation, you're going to trigger some T cells and B cells. I don't know.
I tend to agree with you. So, I mean, I'm sort of arguing a philosophical point here, but I do think that the way that we understand dendritic cell activation is very incomplete at the moment. I think vaccine biologists, I guess I count myself among them, have been looking for 15 years now, looking at different ways to activate dendritic cells so that those dendritic cells, even in an artificial vaccination setting, sort of feel like they had encountered the actual pathogen. And so I think that our understanding of how a dendritic cell gets activated and how it integrates all of these signals and instructs T cells as far as like what the nature of the response should look like, I do think that it's incomplete. And what I want to do to test that is take this woman's blood and pull the serum out and just inject like 45 people with it and then make them all eat kiwi and see what happens. That's what I want to do. Past an IRB. But I think a more likely scenario is that it's one of the cells transferred, right? And I think that this is certainly what the authors believe. And so getting back to my hypothetical mouse with memory T cell repertoire versus memory B cell repertoire, what's your sense of who's carrying the allergy from the donor to the recipient?
Well, I think that the most obvious idea is that kiwi specific B cell that's been previously activated and has a memory phenotype, that B cell made it over into the recipient and can now be activated every time it sees kiwi. It's secreting antibody at some steady state level, and that's getting on the mast cells and basophils of the recipient. That's the most parsimonious explanation, I think. But I think your point about a T cell, if you have a Th2 cell that comes across that recognizes some kiwi peptide and is able to activate B cells that have kiwi-specific antibodies that are naive. The trouble is I'm not even sure that the paradigm that we have of T cells directly in. So, okay, let me back up for a second. Say, in a Th1-driven response, so like an antiviral sort of run-of-the-mill immune response, the way that we think about B cell activation happening is the B cell receptor, which is IgM, that's the first isotype that that B cells make. IgM, bound to the surface of the B cell, binds to its antigen, gets internalized, the antigen gets degraded in an endosome of a B cell, then it gets presented on MHC class 2, and then a T helper cell that is specific for a peptide from that antigen then activates the B cell and provides it with cytokines that cause it to differentiate, usually into IgG, right?
Right.
Okay. So that sort of paradigm, I'm not sure, I can't think of seeing evidence that it works exactly that way in Th2-driven immunity, right? Where it's the same, like, I'm a B cell, naive B cell, I grab my antigen, I pull it in, degrade it, present a peptide on an MHC2, and then a Th2 cell comes in and drives my differentiation into IgE. So there was...
I mean, are you arguing that you don't need T cell help for an anti-allergy response?
You certainly need T cell help, but the sort of like direct contact co-stimulation model, I'm not sure is necessary, particularly for allergic responses. If I recall correctly, again, harkening back to Dan telling me about this, and we should say that both Matt and I have admitted that we basically don't know anything about Th2 immunity.
Right.
Because nobody tells us anything when we take classes in this. But my recollection is that he suspected that potentially these cells were getting stimulated by the protease activity of the antigen itself.
Yeah, so, I mean, the basic outline of that paper is that papain functions as a Th2 driving adjuvant, right? So if you mix papain with any random protein, you'll get a good response to the protein, and specifically that protein will generate a Th2 response. So you'll get a lot of IgA and a lot of IgE. And so there's been a lot of question as to why papain would function in that way, right? And so essentially the outline of that paper is that papain, when you vaccinate with it, either intramuscularly or subcutaneously, finds its way to the lymph node pretty quickly, like basically anything that you inject into the calf or subcutaneously or into the muscle. So it makes its way to the lymph node, and what he could see was that papain would find its way onto B cells, and actually argues that it found its way into B cells, and this is in a receptor-independent fashion. So it didn't have to be a B cell that was specific for papain, it was really any random B cell. And so these B cells would sort of endocytose papain, and through some unknown function modified that B cell's behavior to drive it towards a Th2 mediated response. So he was arguing that the papain's function was actually directly acting on the B cell. Others in that field have suggested that that's not entirely true, that papain and other proteases work on the dendritic cells, and the dendritic cells are really what's going to educate the entire system. As with most, I think, complex scientific things, I suspect it'll be probably a combination of both, but there definitely does seem to be this weird action of papain both on B cells and DCs. And basophils. So there was an argument for a while that what was really happening was that papain was causing basophils to present antigen to T cells, and that's why basophils were important.
That argument.
And that was probably a big lie, and not intentionally. You know, it's just one of those things where it sounded like a really cool idea, and there is evidence that you get basophils.
There were like 3 papers published within like 2 months.
Yeah, and
saying like basophils are antigen presenting cells, and then the next one is like basophils are not antigen presenting cells, and we should do those papers on Audiommunity sometime.
Yeah, it would be fun.
Maybe get Dan to come talk with us.
That would be great, actually. Yeah, we should do that. And yeah, it's definitely not a case of fraud or anything like that. And I think that there was a lot of circumstantial evidence to suggest that these cells were capable of antigen presentation. You know, you find them in the lymph node. They seemed to have MHC. You know, you could get some in vitro stimulation, things like that. So it's not as if they were just like 100% dead wrong in
right. And I think the models that they were using like generated some artifacts that were, if you really understood the system, you might have suspected that was the case, but they didn't fully understand the system, and there was some, like, I think it was like a leaky Cree promoter or something. But
yeah, there
anyway,
were some issues.
That's off in the weeds a little bit. But okay, so we agree that potentially a T cell, maybe a B cell, maybe both could have caused this, could have been transferred and caused this allergy.
I almost think it would have to be. to be honest with you. I almost think it would have to be. And the reason that I think that is that unless you've got, and it could very well be, you've got like a pumping plasma cell in the bone marrow, right? So let's assume that there is an active plasma cell in the bone marrow, or maybe a resting plasma cell. These are guys that are just going to be pumping out antibody. That's their sole purpose in life. So let's assume that you've got one of those in the bone marrow or several of the sister, and then you throw it into a new system. And let's say it freaks out because you just pulled it out of its bone marrow niche and you threw it into the blood of, a random host. And so let's say it gets turned on and you start producing tons of IgE. Maybe that's enough. Maybe that IgE is enough to decorate enough things. But that scenario would argue that just the decoration of mast cells at the site of contact. So if I put IgE in the mouth of him, that would be enough to feed forward into a full-blown allergy response, right? Unless I also, in that same lymph node, had a T cell that was predisposed to respond against this thing.
Well, I'm not sure. If you have a memory plasma cell or a memory B cell, that, I guess those are contradictory terms, memory and plasma cell. But if you have a memory B cell, its niches in the bone marrow,
yep,
it's going to be pumping out, as long as it gets the survival signals, which are just, as far as I'm aware, just some cytokine. It doesn't need continual T cell interaction.
No, it doesn't. It
just needs survival cytokine. So it could hang out in the bone marrow for decades, pumping out some low level of antibody.
Yeah.
Right?
Yeah, so you're arguing that there is no actual feed forward, that this guy has not actually generated any sort of new immune response. What he's experiencing is the vestige of the previous immune host.
What I'm saying is that the thing that drives the pathogenic phenotype of an allergy is IgE on the surface of effector cells. And if you have a memory cell that is secreting antibody, I think that is sufficient to drive a pathogenic allergic response.
Yeah, I guess I am less convinced that the number of cells that you would have transferred will give you some sort of lifelong allergy without some sort of new expansion of those cells. And it's, I have to say here that as far as memory B cells are concerned, we talk about them hanging out in the bone marrow. Like the canonical understanding of how B cells work is that they start in the lymph node and then they go through this germinal center reaction. So you get sort of this survival of the fittest, whichever B cells attack the pathogen best, great. And then they leave the lymph node and then they go to the bone marrow. And there they die or don't die or whatever. It is totally unclear as far as whether memory B cells can recirculate. So do you get, for example, a kiwi-specific B cell that jumps out of the bone marrow back into circulation, finds its way into the lymph node around the mouth area, whatever draining lymph node is in a human mouth, and then sees an antigen, starts proliferating like crazy again, and then generates a new response. So
how much IgE do you need on the surface of a basophil? So you just need 2 to cross-link, right?
Sure, but I would expect that my gut tells me that this person is not just riding a response off the back of a previous stimulation. My gut tells me that you're getting a new immune response.
Well, your gut is scientifically useless.
That is true.
Let's maybe talk about what experiment could we do to determine which of these is true. Because I think it's plausible that it's a T cell or it's a B cell.
Right.
So the experiment, they actually do something akin to this. They want to go check to make sure that this guy's entire immune system is from his sister. And it's actually fairly straightforward because he's a guy and she's a woman, that they basically can just look at the cells and see if they have a Y chromosome or not. And they basically look at this guy's bone marrow and they cannot find a Y chromosome anywhere in any bone marrow-derived cell. Right?
Do they?
Well,
they show this FISH analysis of which there are 4 cells, and then that's all I get.
Sure. Okay, so you could maybe do this a little bit more systematically, but I will trust them that they didn't hide 50%.
Oh no, I'm just not convinced that they did it.
Okay, fine.
But anyway, it doesn't matter because I wouldn't. This guy clearly didn't have a kiwi allergy. Now he does. So clearly, it is a function of this woman's immune system somehow, right?
Right. So the way that we could test, the way that we could find out whether it was a B cell or a T cell or both that was transferred, is actually not trivial. It is not an easy task. Even if you know what the antigen is, it is not an easy task to go sort through an organism's, particularly a human organism's, immune system to try to find the cell that is producing the antibody or that has the T cell receptor, right? This is not an easy thing to find.
It's very hard.
So
it's very hard.
In principle, we could pull all of this guy's bone marrow back out. We could sort out all the B cells, and we could single cell clone that. We could basically put one B cell at a time into a well, and then we could trigger all those wells with kiwi and see if we got an antibody that cross-reacted. But that is not a trivial experiment. That's really hard to do.
Sure.
But if we could do that, then you could look at that B cell and say, does it have a Y chromosome or not? Although actually, no, you couldn't do that. You would have to pull out the same B cell from the guy and go do the same thing in the woman, and then look at the B cell receptor to see if it was genetically identical.
Yeah, here's a better experiment, I think, and I'm going to go back to my human...
my experiment.
No, your experiment's fine. It would just be a horrible, horrible nightmare. So it would...
Not to mention we have to pull out the bone marrow of a guy who's already had a bone marrow transplant.
Yeah, so
and had cancer,
if we could
so that's
not
probably
do
not
that.
going to happen.
Right. So if instead we pulled out a bunch of bone marrow from a kiwi... So mine is also totally unethical. So let's just throw that out there because I'm trying to induce kiwi allergies. I'm looking for sufficiency, right? So here's what we do. We pull out this woman's bone marrow. And the nice thing about hematopoietic stem cells and hematopoietic systems in general is that they propagate. So we can split this out. You could make any number of bone marrow chimeras, right? Especially from a human. So if we suck a pelvis worth of bone marrow out, we could probably get, I would say, at least 100 human immune systems out of that, right? So in the first, let's say 20, we just put in bone marrow and we say, did these people get kiwi allergies? In the next 20, we sort out B cells only, anything with a B cell receptor, those go in. In the next 20, we sort out T cells only, those go in. And the next 20, anything that's not a lymphocyte goes in. Right? Or we just pull the serum again, because I like the serum control, the IgE control. But I think what we really need to do is test what cell compartments are responsible for this. Okay. And then once you have the cell compartment, if it turns out to be the case that transferring B cells is not sufficient to drive this allergy, then this becomes infinitely more complicated, right? Because that means that it's not the antibody itself that's starting it. It's a predisposition of something instructing the antibody development and production.
Yeah, but I'm not convinced that your experiment actually will prove definitively one way or the other.
No, it'll give you a sense of...
Because we're already admitting that it is a small fraction of the B cells in a particular person that are going to have that kiwi response. Small proportion of the T cells.
Sure.
And we're doing our best to purify out in a normal hematopoietic transplant. I know that you're
right. I'm
you're
not
going to
doing
pull
that.
out certain
I'm
things.
going to pump a ton of B cells into each individual recipient. Yeah.
All right. So what if we see that both T cells and B cells are sufficient by themselves? Either one can induce an allergy.
That would be interesting, wouldn't it? It would mean that once you go down this path, like once you establish the allergy, that you have both B cells and T cells that are involved in the maintenance of that allergy, which makes this a much more complicated problem.
It's also possible that one of the reasons that this has never been reported before is that you actually need both the kiwi-specific T cell and the kiwi-specific B cell, because there are tolerance mechanisms built into both B cell and T cell responses that have to go awry in order for you to get an allergy.
Yeah, and
so it's possible that you need both to be transferred. One by itself will be insufficient to direct a new allergy because the tolerance mechanisms in the recipient on either the B cell lineage or the T cell lineage alone would be sufficient to block the development of a new allergy.
That's my favorite theory. As I was reading through this, I think probably there was leakage from multiple cellular compartments, all that combined to turn an allergy into a full-blown thing, and I think that those all were able to ramp themselves up in this new recipient. That's my thought.
I would believe
I like it.
We don't have a way to test this ethically, unfortunately. Maybe we could make a humanized mouse and just inject a bunch of crap into mice.
Would you even need to? You could totally do this experiment with house dust mite.
Maybe.
Yeah. So you take a cohort of mice,
make mice
really allergic to house dust mite, and then start just transferring different cellular components from the bone marrow into recipients and see who gets it.
Yeah, I guess you could maybe do that. It wouldn't tell you what happened in this particular patient, but it would tell you what is possible.
Sure.
Yeah. All right, let's do it.
Yeah.
I don't have a mouse colony. You're on it.
I do. I got some. I got some mice.
All right, report back in 2 weeks.
Sounds good. Actually, that's not a terribly hard experiment to do. I mean, it's not the easiest experiment, but it's not the hardest experiment to do. That could be fun.
All right. I look forward to hearing your report.
Yeah. So let's go make some mice that hate kiwi. I don't hate kiwi.
No, I love kiwi
a lot.
It's delicious.
Yep.
I feel really bad for this dude.
I know.
Although not as bad as I would if he had died because he didn't get a bone marrow transplant.
Yeah, and if this gets back to you, Guy, with transplanted bone marrow, I hope that the leukemia thing is going well, because that's obviously a much more important thing than this ridiculous kiwi shit that we're talking about. So
agreed.
Yeah.
All right. Well, this has been a fun, philosophical, and not particularly productive episode of Audiommunity.
Yeah.
I'm Kevin Bonham with Matt Woodruff and previously Kate Franz. It turns out Kate mentioned Google Play last time that we recorded, and it turns out that you can listen to podcasts on Google Play. And I have submitted us to Google Play, and we are now on the Google Play podcast library. We're also on Stitcher, and we're on iTunes, and we have an RSS feed that you can grab from our website, emmunity.org, emmunity.org. We're also on Facebook, facebook.com/audiomunity. You should find us in all those places. You can also send us comments, either through the form on our website or at comments@emmunity.org. So you got a lot of ways to reach us. Please reach out, because we do read your email responses. We don't get a lot of them, so it doesn't take us a ton of time. So if you get in early, we're going to remember you, and then in the future, when we're famous, we're going to still respond to your emails. That's bull. I don't think that's going to happen. But yeah, we're all over the place on the internet, so go find us, I guess.
And on that note,
we'll see you next time.
Guys.
Music at the beginning and the end of the show was composed by Rachel Reineck, and Rachel Reineck is going to be having a fundraising campaign for her next album coming soon, and I will report that here. And I may change the intro and outro music because her new shit is really, really good, but that's to come. So with that, we'll see you next time.